Thank you very much for the invitation to speak. So I'm going to change subject a little bit now. I'm going to talk much more about modelling, how modelling can be applied to understand the behaviour of simple cells and how that can give rise to much more interesting and complex emergence. So just to start with, I'm going to explain the basis of the technique which I use, which is called agent-based modelling, and how we started off by applying this to rather simple in vitro or experimental systems. After I've done that, I'm then going to give a couple of examples which may be a bit more aligned with the in silico medicine theme. So talk about how we've applied these models more recently to look at colorectal cancer and also looking at fertilisation. Okay, so the basis behind the ideas I use is the idea that complex systems can be made up of quite simple individuals that might obey simple rules and interact quite simply, but still give rise to some quite interesting non-intuitive behaviour. So if you think about a flock of birds, you can think about those birds as being sort of individual entities following simple rules for how they change their direction and speed. But together that flock can have quite interesting behaviour and show some quite unpredictable dynamics. And we can think about cellular systems in the same way. So for instance, how an embryo develops, how a tumour develops, and how maybe a wound heals, which is shown in the slide here. You can think of these systems being made up of simple cells that all interact with each other, but there is no overarching control. So there's no one master cell or God in the system, if you like, which tells each individual what to do. So in order to capture the interesting and important behaviour of this system, we need to represent the individuals in this system as individual entities. So individuals and their differences are important. So the idea behind agent-based model, whatever system you're looking at, whether it's biology or social or transport, is that you have this virtual environment which contains the things that you're interested in, as well as a set of virtual entities, which in my case represent individual cells. Each of these entities has a state, and then you pre-program your rules based on the understanding of how these entities interact and behave. And these entities then update their state, and this is done iteratively over many, many time steps. So one of the early studies we did to look at this is to use this paradigm to understand how cells in a monolayer behaved, and then if you created a wound in this monolayer, how that depended on the behaviour of the cells and the condition of the environment. So we looked at two examples, a system where there was a normal level of calcium, which meant the cells were quite sticky, and a very low or artificially low level of calcium where the cells weren't sticky. And then we ran simulations based on very, very simple pre-programmed rules to see what happened. They weren't synced, unfortunately, but what you can see is very different qualitative behaviour in how the two wounds heal. In the sticky wound on the left, there's a slight movement of complement sheets. In the non-sticky wound on the right, the cells migrate in much more quickly to close the gap. So one of the advantages of a system like this is that you can run a directly analogous set of experiments in your lab to see if your model is predicting the correct things. So these were experiments done on urothelial or bladder epithelial cell monolayers by my colleagues at the University of York. And what you could see is the same qualitative type of behaviour. So this was a proof of concept, if you like, that this kind of approach was applicable to modelling populations of cells. And although you may not see the direct analogy to the medical problems you might be interested in, these systems are interesting in terms of understanding proliferative control and homeostasis and response to injury. And they're also very easy to image and get data from compared to the in vivo systems, which I'm now going to move on to. So much more recently, we've been looking at understanding colorectal cancer and how it arises. So it's important because it's the second largest cancer killer, both in Europe and indeed worldwide. And what happens is you get what's called adenocarcinoma, which is a cancerous region. And that's surrounded by a precancerous spreading field, which is usually associated with a particular mutation in cells called an APC mutation. So the colon is made up of these structures called crypts. So this here is an example of around about 10 crypts. And the cells undergo proliferation and division at the bottom of the crypt, and then they are pushed up towards the top. But how these mutations then spread through the colon is poorly understood. So there's a hypothesis that it might be due to cells from a neighbouring mutated crypt spreading down into the crypts on either side, or cells that have undergone a mutation can then undergo fission and split into more crypts. And this is very, very hard to study in vivo. So we had a student looking at the system. So he developed, first of all, a model of a single crypt with cells with an APC mutation at the bottom. And you can see how these cells then divide and spread up and take over the whole crypt. And he actually extended this for the first time into a model which contained multiple crypts. And you can see this green spreading field associated with those cancerous or precancerous cells. What we never observed under any parameter set that we put into the model was the case where the green cells actually invaded the crypts of the neighbouring crypts. So this suggested support for the fission hypothesis of field spread. So with this model, we were able to model up to 25 crypts for a period of round about a year. Okay, but precancerous changes in the colon can take place over a period of decades, and there's around about 5 million crypts in the colon. And we worked out if we used a simulation to model that would take 15,000 years of compute time. Okay, so not something that was going to be feasible. So what we then did was look into building a second scale model. So what you're looking at now is a fixed picture, it's not meant to move, where each of the dots you can see represents not an individual cell, but an individual crypt and the flat mucosa associated with that crypt. So by using information that we gained from the cellular level model, we were able to inform rules for the crypt level model and look at much larger simulations. So for those of you who are modelers, this is an example of a decoupled multi-scale model. Okay, just moving on, just to give you a flavour of another example where we've developed computational models. And this is actually an area of medicine which has been relatively untouched by the computational modelling community compared to areas such as cardiovascular medicine and cancer oncology. There are very, very few models that you'll find that look at fertility. Okay, so I think that's a reflection of the fact that fertility issues don't necessarily kill people, and the funding in this area is probably a bit less than you would get with other medical fields. But it is a growing problem. It affects 1 in 7 couples in the UK, and it's a complex biological process that involves interactions between cells from both the maternal and the paternal side, the ova and the sperm, and also a very, very complex maternal environment in terms of the oviduct, which is what we know as the fallopian tube in humans. So basically the system our model tries to catch is that you have a fallopian tube, you have a reservoir of sperm, which are attached at what's called the UTJ, the uterotubular junction, and then you have oocytes or eggs, which are released by the ovaries and move down in the opposite direction. And then hopefully what will happen is that population of sperm will move up the oviduct, interacting in quite complex ways with the maternal environment. Hopefully some of them will reach the oocyte and penetration will take place. So this is a nice, relatively straight image here. Unfortunately, real oviducts don't look anything like this in real life. So this is much more representative of what a real mammalian oviduct looks like. And this was based on a 3D reconstruction from micro-CT images taken from mice. And hopefully this is going to run. Okay, so this was the environment for our agent model. The agents in this case are sperm, which start off at the UTJ. They become what's called capacitated. They change their motility behaviour, and then they gradually move up through this environment towards what's called the ampulla, the oviduct, at the ovary end of the oviduct, and the ova are represented by these pink circles here, and then hopefully what you will see is that eventually at least one of the sperm agents will reach the ova, and at this point the simulation stops. The inside of the oviduct is also much more complicated than a simple tube, so I am hoping this is going to run. So this is a reconstruction, this time from histology images from the mouse oviduct, which are linked together. And what you can see here is essentially a sperm's eye view, sort of swimming up the oviduct. So I don't think it gets to the point where it meets an egg and fertilization takes place, but it just gives you the idea of the complexity of these environments. So as well as the computational modeling challenge, there's also the graphics challenge that goes with these type of simulations. Oh, and here's an example of the histology images which were used to recreate this internal environment. So this presentation has been quite high on interesting movies, possibly a bit low on the scientific side, but just to convince you, hopefully, that we have considered validation. This is an attempt at validation of this fertilization model. So you can see the simulation results in the blue line compared with real data from animal experiments, which are shown in red. So for the majority of the time points that we're looking at, there's very, very close agreement between the experimental data and the computer simulations. What I would say is that this data that we're validated against is from the 1950s, and that's the only data set that we could get hold of. So there's a very big difficulty in terms of obtaining the data to validate models against. I'm not going to talk about multiple myeloma, which is another ongoing project, simply because I've run out of time. Okay, so just to summarise, agent-based modelling has been used in simulating both in vitro and in vivo systems. There's ongoing work, so projects I haven't talked about, modelling stem and cancer cell interactions in order to look at potential drug delivery systems, modelling endothelium or blood vessel tissue, and also the multi-scale modelling component, which I've only just touched on. There are lots of challenges with this work. One of the main ones is getting good data in order to inform and validate models. The other thing is that, obviously, what you can appreciate from these movies is that there's an opportunity here to explore the educational and outreach side. So in terms of the fertilization work in particular, we've got a project about to start supported by Insigneo. It's a summer internship project to develop a 3D VR tool for these oviduct simulations to help educate the public and also medical students. Okay, so just my acknowledgements finally for the various groups that have been involved in these projects. Just to point out that for the fertilization modelling, my PhD student, Carlos Morales Garduno, has a poster which you might want to look at. It's number 33. And for the myeloma work, which sadly I didn't have time to talk about, another student, Curtis, has his poster, which is number 43. Okay, thank you very much for your attention.
Thank you very much, Dawn, and thank you to all our speakers for excellent timing. So do we have any questions for Dawn?
Nice talk, Dawn, and very nice visuals.
Thank you.
Yeah, so I see the potential of this agent-based modeling being able to model like the heterogeneity of cells.
Yes.
Right. So I was just wondering, like, how do you model each cell with each agent with like different attributes, and how do you train that model?
Absolutely. So that's one of the major strengths of agent-based modelling, that you can capture the variation or heterogeneity in different cell populations. So you could look at normal cells versus mutated cells, for instance. So that's done to some extent in most of the models that I presented. By training, do you mean calibration?
Yeah, I
So-
guess do you hard code those rules, or do you actually
Yeah.
train on the data, some observed data?
Yeah, so it's kind of an iterative process. So you start off by putting in rules and numbers which you think are reasonable. You build a simulation, and then hopefully you get hold of a reasonable data set. And you then identify which parameters are the most important in controlling the system and which are the least well known, which is a process called sensitivity analysis. And then you can undergo a process of essentially calibrating or tuning those parameters in order to make the model fit the data. So that's something where we haven't used machine learning, but I know there's ongoing work in certainly Curtis's work on the myeloma and other projects that is related to his, which are looking at interfacing with machine learning techniques to try and do this calibration.
Yeah, and there's also like interesting work coming out from single cell technologies that you could use to like train your.
Yes, so the
train
more
your
single
models.
cell data we can get hold of, the better, definitely.
Okay, so that's the close of this session. Please enjoy the coffee break and don't forget to visit our exhibition while you're having your coffee.